So it's almost as if QTLs are a subset in a way of key risk indicators. They're ones that focus on the really critical areas. They're also their study level, so that you don't have QTLs at a country level or a site level. They're very much at a study level, because it's all about whether the study itself is going to succeed or not, at least from an operational perspective, not in terms of efficacy and safety. So they're study level, and you can certainly have study specific quality tolerance limits, but often there may be QTLs that are used commonly across a program of studies. So for one molecule, for instance. And why might that be the case? Well, because these are focused on what's really important, what really matters for the individual studies, and then overall, actually, if you think about it, for the overall program. And so there are likely to be some QTLs that actually are common across studies. They're determined at a program level and then used across all of the relevant studies. And they're focused then on features of the study that are critical to study success. That's that critical to quality. That's again language out of ICH E6 R2. So examples here, things like the withdrawal rate of participants, whether endpoint data, critical endpoint data is missing, on randomization or stratification in double-blinded trials. And so these, of course, you could see, if these are wrong, that could be very critical. If the withdrawal rate of participants in a particular study, for instance, is particularly high, so you don't get all of the data that you need per participant, the question is always going to be, well, why did those participants withdraw then? And is it actually because there was a problem with the study, with the drug, or whatever? And it's an unknown. It suddenly becomes an unknown, as of course does missing critical endpoint data becomes an unknown. So as you can see, as I've sort of described those, they would typically then be defined with both medical and statistical expertise. So the statistician will be able to say, if this particular data is missing, or this proportion of it is missing, or if we don't have enough participants, or this proportion of participants withdrawal, then we're likely to have a problem, and in particular a problem in submitting it to a regulator. And those levels, if possible, the levels of the quality tolerance limits, so at what point it becomes a concern, should be based on historic data, but you don't always have historic data. And I think the industry is still in early days in implementing these. But you should have quality tolerance limits, and as I say, if you think of them as sort of special, elevated, critical, if you will, key risk indicators, special key risk indicators, and you've already got key risk indicators, then this is almost like having some of those that you elevate into that very critical sort of level. And as a reminder, it's in the regulation, deviations from the quality tolerance limits have to be reported in the clinical study report.